The overall goal of this procedure is to generate cord blood-derived cytotoxic T lymphocytes, or CTL, that recognize antigens from multiple viruses. This is accomplished by first isolating the cord blood mononuclear cells from the 20% fraction of the cord blood unit. The second step is to use the mononuclear cells to generate dendritic cells, Epstein-Barr virus lymphoblastoid cell lines, or EBV-LCL, and the virus-specific CTL. The mononuclear cells, which contain the T cells, are stimulated with cells that have been transduced with the Ad5F35PP65 vector. The final step is to confirm the specificity of the multivirus-specific CTL and to freeze the CTL. Ultimately, the specificity of the expansion of the CTL for at least one viral antigen can be evaluated through ELISpot analysis and cytotoxicity assays.
Generally, individuals new to this method will struggle because the volume of cord blood available in the 20% fraction is very small, which means that there are very few cells to work with. The procedure is also quite intricate and involves the generation of at least 3 different cell types from these limited cell numbers.
We first had the idea for this method after we analysed the data from a clinical trial where we made multivirus-specific T cells for patients after bone marrow transplant. In that study, we saw that the T cells were not only safe but efficacious in these patients and prevented and treated viral infection. However, the patient population who have the greatest need for such a therapy are recipients of cord blood transplants. In these patients, however, we are unable to go back to the donor to get blood to make T cells. So we had to develop a novel strategy to